Okay, so enzyme helps the food to digest, help to digest food in humans and are produced by different parts of the alimentary canal. The box on the left containing the sentence began. Okay, draw two lines to make the correct sentence about enzyme. What are enzymes? Good. So, oh, sorry, I have made the incorrect one line. Wait, sorry. So it's not like that. So the first one is they are catalysts that speed up the reaction and are unchanged. So which one is that? This one? And the second one would be this one, right? Let me change the pen. Okay. The next question is figure 3.1 is a diagram showing an enzyme and several different substrate molecules. What is the correct substrate for this one? Yes, good. Okay. Now there is a graph, there is a table. Let's see what we can do about this. We have directly moved to deep. Some questions, some questions deep part. Skipping the C one. Okay. So here the enzymes are given with different pH, which works at different pH. Okay. Now the question is the pH values in different part of the alimentary canal were measured. The use the information and tell which enzymes would come over here. What would be the enzyme of duodenum? Okay, the duodenum the pH value is 5.5. In ileum it's 8. In mouth it's 6.7. In stomach it's 1.5. So according to these graphs you have to name these U, V, W, and X. Okay, the first one 5.5 is yeah. No, no, just wait. Okay. First one is 1.5. So which one is this one? Where should we write U? Okay. V kahan? Duodenum. Okay. Then W, 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 mouth. Okay. And X, ileum. Okay. Name one part of the large intestine. What do we call it? Colon. We call it colon. Okay. I'm giving you more questions. Wait. There is one question related to pectinase. Now, pectinase is part of the exactly the you must have read this in biotechnology, but definitely it's an enzyme, so it's part of enzyme as well. Pectin is an enzyme that is used in the production of apple juice. A student investigated how affected the volume of juice produced when using pectinase. The student chopped the small apples into small pieces, and then pectinase was added. After 2 hours this is observed. This is the volume of juice at different pH. Now what do you think, which is the optimum pH and why? No, you have to give one value. You have to give one value. When it's pH, you have to give one value. Where is the value highest? Where is the highest volume? At 5 pH. And what is the reason that you have selected this? No. What is the reason? Why do you think 5 is the answer? Good. It produces the highest volume of apple juice, which is 27.6 cubic centimeter. Okay, you have to like give the complete value. Whatever is asked in the question, use the terms from the question. Okay, now there is another graph related to pH, and the question is describe the effect of this pH. Okay, the question are mostly about the pH instead of temperature. Okay, Describe the effect of pH in increase on the rate of reaction. Now you, in description you have to give all the values from where the activity started, where it's maximum and where it's the lowest. Okay. No, no, no. It's not important. Just mention the important terms, important ones. Wait. I think so. My phone, my laptop is not charging. There is an error. Wait, wait, wait. I'll explain. Uh, what has happened? OMG. If I got logged off, you need to understand that my laptop's battery is out. I don't know why it's not charging. The port is not working. Let's bear with it. जैसे भी चल रहा है इसको चलने दे चल रहा है ना okay 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 so first of all we'll start from the starting point. This is our point number 1. When I say point number 1, it means I'm talking about mark. This is mark number 1. Then I have to tell about the general description. This is my point number 2. Point number 3 is the maximum one, and where it drops back to 0 is my point number 4. This is how you got your 4 mark. Okay, let's say I'm just answering it. So at pH 5 the activity before pH 5 the activity is 0, but it increases exponentially. Now I have use the word exponentially. Exponential mean very faster. Is my screen visible? Okay, yes, exponentially means a very fast or very like high increase, rapid increase basically. Where at pH 8.3 the activity is maximum, which is 6 AU. Okay, beyond this pH the activity decreases to 0 at 10th. Okay, or the rate of reaction becomes 0 at 10 pH. This is how you answer the like 4 mark question. Whenever explain the graph, give a general trend, give a certain, and then give a value where there is a sudden release or sudden decrease. Okay, tell the maximum value, tell what happens after that maximum value. This is how you do it. Okay, this was अगर आपके पास topical है, you may find this question in your topicals. Okay, a student investigated the effect of lactase on three different liquids. Okay, what is lactase? Which breaks down milk, basically the lactose sugar. First thing is milk. Second one is lactose free milk. Third one is sucrose sugar. Okay, now lactase is going to act on milk, not here and not here. Lactose free means there is no lactose, then there is no meaning of lactose is not going to act on lactose free thing. Is it so? Okay, then we have used some indicator to check the presence of glucose, whether glucose is present or not. The indicator turned brown in the presence of glucose. Okay, the results are given. Okay, in milk it becomes brown after treatment with lactase. In lactose free it is brown in both cases, but in sucrose it is not. Why do you think? Why do you think so? Why? They are already converted into glucose. They're already converted into glucose. Wait, wait, wait. Wait. Listen. No, I'm telling you some like story. You need to listen. Okay. We have three different solutions. In the first one, there is milk. Okay. Now, milk doesn't contain glucose. It contains lactose. Okay. When we add an enzyme, it breaks down into glucose. Okay. So your result number one is before treatment it is blue, but after treatment it is brown. Means before treatment there is no glucose, but after treatment the lactose is converted into glucose. ठीक है. Now second one, lactose free milk means it is already treated. Lactose free is already treated with lactase. So how it is made, even in industries nowadays it's very common lactose free milk because people are allergic to lactose. So in industries they have already used the lactase enzyme and convert the lactose into glucose. That's why the result is positive even before and after the experiment in both cases. Okay. And in third one, where sucrose has nothing to do with lactase, sucrose is definitely going to be broken down with the help of sucrase enzyme, not lactase enzyme. That's why the result is negative before treatment and after treatment. Now you have to explain this result in your answer. Yes, in lactose free milk. Look, it's an industrial process, na? The lactose free milk is produced in the industries. It's already treated. It is already treated with lactase. So lactose is already converted into glucose. Glucose is present. That's why the color is brown before the treatment and after the treatment. जी 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 मैं बता सकती हूँ. समझाना है या लिखवाना है?